Whenever you see this sign in a biology laboratory, you know that radioactive materials are being used. Many of them can be detected with a Geiger counter. The more clicks the counter gives, the more radiation is being given off. Radiation is given off by radioisotopes. The atoms of most chemical elements occur in several different forms or isotopes. Carbon, for example, has isotopes of mass number 12, 13, and 14. Sometimes one of the isotopes of an element is unstable and breaks down, giving off radiation. This form of the element is called a radioisotope. Isotopes which are radioactive obviously have different physical properties from non-radioactive isotopes, a higher mass number, for example, but they have exactly the same chemical properties. This makes them one of the most powerful tools available to biologists and biochemists. A living cell can't distinguish between the radioactive and non-radioactive isotopes of an element. So radioisotopes can be used to tag or label molecules. This means replacing some of the atoms in the molecule with radioactive atoms, so that the molecule can be followed wherever it goes in the cell or the organism. Its presence is detected by the radiation it gives off. And these days, very tiny amounts of radioactivity can be detected and measured very accurately. There are a number of ways of detecting radioactivity. It will blacken photographic film, for example, and this property is often used in biological experiments to produce autoradiographs. This chromatogram's been run in solvent, dried, and then sprayed with a color reagent. This revealed the position of the sugars, but doesn't show which ones are radioactive. To find this out, the chromatogram is taken into the darkroom and put next to a photographic film between two sheets of glass. It's left there for a few days, and then the film is developed to produce an autoradiograph. Wherever there was radioactivity on the paper, the film is blackened, so it's easy to tell which of these sugars is radioactive. This blackening of photographic film by radiation is also used as a safety check for people who work in laboratories where radioactive materials are used. Inside these film badges, which everyone in the laboratory must wear, there are little pieces of photographic film. At monthly intervals, the film is replaced and the old film developed. The blackness of the film is an indication of how much radiation the person has been exposed to. This approach is useful for detecting radiation in general, but it's not sufficiently accurate to give good quantitative measurements. More accurate methods for measuring radiation depend on the type of radiation being emitted. The three isotopes most commonly used in biology are P32, C14, and H3. These all emit beta radiation, streams of high-energy, fast-moving electrons. One of the simplest ways of detecting beta radiation is with a Geiger counter. The Geiger-Müller tube, named after its original designers, is an ionization chamber, a gas-filled tube containing an anode and a cathode. with a high potential difference between them. When a charged particle, like an electron, passes between the two electrodes, there's a transient increase in the conductivity of the gas, which gives a pulse of electricity. This pulse is fed to a loudspeaker to give the clicking sounds, and recorded as counts. Quantities of radiation are therefore often expressed as counts per second or counts per minute. The more counts there are in a given time, the more radiation is present. But the number of counts recorded is not the same as the number of radioactive decays in the sample of isotope. This is because this type of equipment isn't very efficient at detecting every radioactive disintegration or decay that takes place near it and recording it as a count. For instance, although a radioactive substance emits radiation in all directions, only some of it will be directed towards the Geiger tube and trigger a discharge. The rest may go undetected. When one radiation particle has caused a discharge, a finite time has to elapse before another can be detected. So, at high levels of radioactivity, a significant number of radioactive disintegrations taking place will not be recorded as counts. In addition, it's almost impossible to use a Geiger counter for measuring tritium, which only gives out relatively low-energy electrons. So instruments called liquid scintillation counters have been developed in order to increase the sensitivity and make the very accurate measurements of beta radiation that are necessary in modern biology experiments. You'll find these in biology and biochemistry laboratories all over the world, and the principle on which they work is very easy to understand. When certain organic chemicals are bombarded with beta particles, they emit light. So, if a radioactive compound and one of these chemicals are mixed together in a suitable solvent, the radiation given off by the radioactive compound will cause the organic chemical to give off flashes of light. A liquid scintillation counter is a machine which can count the number of light flashes, and the more light flashes there are in a sample being counted, the more radioactive compound is present. These samples are ready for counting in the machine. Each glass vial contains the radioactive compound and the special organic compound in its solvent. When the sample is taken down into the machine, the light flashes are detected by a photomultiplier tube, and each flash of light is recorded as a count. The number of counts per minute being recorded is shown here during the counting and can be printed out at the end. The photomultiplier tube in the scintillation counter responds much more rapidly than the tube in a Geiger counter. It can also detect a very high proportion of all radioactive disintegrations that take place in the sample, so even extremely low levels of radiation can be measured. Radioactive isotopes are used by biologists to study many of the cell's vital processes. One of these is protein synthesis. When a cell makes proteins, the genetic information stored in DNA is carried out into the cytoplasm by messenger RNA to the ribosomes. There, amino acids brought to the ribosomes by transfer RNA molecules are assembled into proteins. These are the red cells of the blood, the erythrocytes. In each of these mature red blood cells, there are nearly 300 million molecules of hemoglobin, the protein that carries oxygen from the lungs to the tissues. All this hemoglobin is made by an immature form of the red blood cell called a reticulocyte. Unlike the mature cell, the reticulocyte has a nucleus, so it has DNA and all the information for making proteins. This tube contains a suspension of reticulocytes from a rabbit. It's red because the cells contain hemoglobin. You can't see the individual cells, of course, they're much too small. The cytoplasm of these cells contains everything necessary for making hemoglobin: messenger RNA, ribosomes, amino acids, transfer RNA molecules, and a source of energy. So it makes a good experimental system for demonstrating protein synthesis. These reticulocytes are being kept very cold at the moment to preserve the structure of the delicate enzymes and to slow their activities right down. The demonstration is easier to do if the outer membranes are removed from the cells. This ensures that anything added to the system is added directly to the cytoplasm and the protein synthesizing system. To remove the membranes, distilled water is added to the cells. The water is drawn into the cells by osmotic pressure and bursts the cell membranes. As they burst, the cell contents are released. The fragments of cell membranes are removed by spinning the tube in a refrigerated centrifuge. The membranes pack to the bottom, so the protein synthesizing system of the cytoplasm can be gently separated into another tube. Now, protein synthesis can't be demonstrated in a system like this by weighing the amounts of protein present at different times, because any increase is far too small. But protein synthesis can be demonstrated using a radioisotope. If a radioactively labeled amino acid is added to a protein synthesizing system, it's incorporated into the growing protein chains on the ribosome. So the proteins which are made in the following minutes become radioactive. The radioactive label allows the newly synthesized protein to be detected, even if only a tiny amount is present. The appearance of radioactive protein in this system would be proof that protein synthesis was taking place. So all that's necessary is to add a radioactively labeled amino acid, take samples from the mixture during the following minutes, separate the protein from the samples, and then show that the protein is radioactive. To be sure that any radioactivity detected in this last step is from radioactive protein, the protein must be separated from the sample in a way that ensures that there are no radioactive amino acids still present. This can be done very simply. These two tubes contain some acid. If amino acids are added to the tube on the left, there's no visible change. If protein is added to the tube on the right, the protein precipitates. So, adding a mixture of amino acids and proteins to acid will make the proteins precipitate, but leave the amino acids in solution. In the same way, acid is used in the experiment. to separate radioactive proteins formed during the experiment from any radioactive amino acids which may remain in the cytoplasm. Going back to the main experiment, some of the protein synthesizing system that's been kept cold is put into a water bath to bring its temperature back to 37 degrees centigrade, body temperature. To start the experiment, a small amount of a radioactive amino acid is added and the contents of the tube carefully mixed. After 5 minutes, a small sample is taken out and added to cold acid. With acid at this strength, all the proteins in the cytoplasm should be precipitated, including the enzymes, so no more protein synthesis should take place in this sample. But keeping the tube in ice is another way of ensuring that there are no further enzyme-catalyzed reactions. 5 minutes later, a second sample is taken from the system and added to acid. And this process is repeated every 10 minutes until there are 6 samples. Finally, some acid is added to the main tube to stop any further protein synthesis in the bulk of the reaction mixture. To collect the protein from this sample, remember that's the precipitate, the sample is filtered through a little piece of filter paper. It's washed well, and then dried, first with ethanol, and finally with acetone. The same process is repeated for the other 5 samples. To find out if this dried protein contains any radioactivity, it must be put into a vial with some scintillation fluid and counted in a scintillation counter. Radiation from the radioactive protein will cause the organic chemicals in the scintillation fluid to give off light. This will be detected by the scintillation counter and recorded as counts per minute. Each of the 6 samples is going to be counted for 4 minutes, and once the machine has been set and started, it runs automatically, counting each sample in turn and printing out the results. It's obvious that all the samples were radioactive, and the amount of radioactivity increased with time. There's no doubt at all that the experimental system from the rabbit reticulocytes was synthesizing protein, but it wouldn't have been possible to show that without using a radioactive isotope. In any living cell, there are thousands of different proteins, some forming part of the cell's structure, some responsible for its complex functions. All proteins are chains of amino acids, and the chains of each type of protein have a unique three-dimensional shape. It's the sequence of amino acids in a protein chain that determines how that chain will coil and fold up. And for every type of protein, that sequence is different. To keep functioning properly, a cell must be able to replace or renew its proteins. When it's growing or dividing, it must be able to make new proteins. The information that allows the cell to do this is stored in the nucleus in coded form in the molecules of DNA. Each strand of a DNA double helix is a chain of nucleotides. Each nucleotide is made up of a base, sugar, and phosphate. The chains contain 4 different bases, either cytosine, C, guanine, G, adenine, A, or thymine, T. And it's the sequence of these bases in DNA that contains the information for the sequence of amino acids in proteins. The information is in the form of a triplet code. In other words, a sequence of 3 bases is the code for one amino acid. So, if you know the sequence of 3 bases for each amino acid, you can interpret the sequence of bases in a length of DNA as the sequence of amino acids in a protein. In this example, it would be alanine, serine, lysine, and so on. DNA stores the instructions for protein synthesis, but before the cell can act on these instructions and make proteins, a number of other things are needed. The information about protein structure, stored in the genetic code of DNA, is in the nucleus. Protein synthesis takes place on tiny organelles called ribosomes, which are in the cytoplasm. A messenger molecule is used to transfer the necessary information from nucleus to cytoplasm. The messenger molecule, messenger RNA, is made in the nucleus on a single strand of DNA, and the sequence of bases in its nucleotide chain is determined by the sequence of bases in the DNA. Wherever there's an adenine in DNA, there's a uracil, U, in messenger RNA. Where there's a thymine in DNA, there's adenine in messenger RNA. Where there's cytosine in DNA, there's guanine, and where there's guanine, cytosine. This is called base pairing, and the 2 bases which form each pair are said to be complementary. As a result of base pairing, the genetic code in DNA gives rise to a complementary code in molecules of messenger RNA, with a sequence of 3 bases, known as a codon, specifying 1 amino acid. It's in this form that the instructions for making a protein are passed to the ribosomes. Ribosomes are made of RNA and protein. The ribosomal RNA consists of a number of RNA molecules of different chain lengths, from several hundred to several thousand nucleotides. These molecules coil and fit together with the ribosomal proteins to form 2 irregularly shaped subunits, one large, one small. Together, these form a structure on which all the various molecules involved in protein synthesis can come together and function correctly. Amino acids are brought to the ribosome by yet another type of RNA called transfer RNA. There's at least one transfer RNA molecule for each amino acid, but they all have the same general structure. Drawn like this, to show how the different parts of the nucleotide chain bond together, a transfer RNA molecule looks a bit like a clover leaf. But in 3 dimensions, it's more like a twisted hairpin. And in a simple form, it can be drawn like this. There are 2 parts of a transfer RNA molecule which are very important. At this end of the molecule, the transfer RNA attaches to its corresponding amino acid. So, if this was the transfer RNA for alanine, alanine would be attached here. If it was the transfer RNA for serine, serine would be attached here, and so on. Attaching an amino acid to its transfer RNA needs energy, from ATP, and specific enzymes which can recognize both the amino acid and the transfer RNA. At the other end of the transfer RNA molecule, there's a sequence of 3 bases which is specific for the amino acid it carries. In the case of alanine, these 3 bases would be CGA. For serine, they would be AGU, and for lysine, UUC. The 3 bases here make up the anticodon, and their sequence is complementary to the sequence of bases making up the codon on messenger RNA. This means that transfer RNA molecules can line up on a messenger RNA molecule, but they can only do so in a certain order. That order is determined by the order of the codons in messenger RNA, which, in turn, is determined by the sequence of bases in DNA, the genetic code. The interaction of codons and anticodons at one end of the transfer RNA molecule ensures that, at the other end, amino acids are lined up to make proteins in the order originally specified by the sequence of bases in DNA. So, the sequence of bases in a length of DNA is first copied or transcribed into messenger RNA and then translated into the sequence of amino acids in a protein. Protein synthesis begins when a messenger RNA sent out from the nucleus links up with 2 subunits of a ribosome and with the transfer RNA carrying the first amino acid of the protein sequence, a process that also needs the help of various initiation factors, soluble proteins from the cytoplasm. In eukaryotic cells, the first amino acid brought to the ribosome is always methionine. This doesn't mean that all protein chains start with methionine. This methionine is usually removed once the protein chain has been made, but it does seem to be necessary to get the process going. The codon for methionine is AUG. This codon is known as the initiation codon, and it marks the start of a protein sequence. When it engages with the anticodon on the transfer RNA for methionine, protein synthesis is set in motion. Now, a ribosome has 2 sites where a transfer RNA molecule can bind. So, 2 transfer RNA molecules can be associated with a ribosome at any one time. These sites are called the P site and the A site. Abbreviations for peptidyl and aminoacyl sites. When protein synthesis is set in motion, the transfer RNA carrying methionine is located at the P site, which is adjacent to the initiation codon AUG. The next codon on the messenger RNA, here it's GCU, is adjacent to the A site. The anticodon to GCU is CGA. Remember, there's complementary base pairing. And the transfer RNA molecule with that anticodon is the one for alanine. So that's the transfer RNA which comes and binds here, bringing alanine alongside methionine. A peptide bond forms between them, joining the first two amino acids of the protein chain. In the process, the bond between methionine and its transfer RNA is broken, and it's the breaking of this bond that supplies the energy to form the peptide bond. Two amino acids are now joined together, but before the next amino acid can be added, two things happen. The transfer RNA, which no longer has an amino acid attached to it, leaves the ribosome so that the P site is vacant, and the whole ribosome moves along the messenger RNA until the transfer RNA, which has the growing protein chain attached to it, occupies the P site, and the A site is adjacent to the next codon on the messenger RNA. In this example, it's UCA. Now the process is repeated. The transfer RNA with the anticodon to UCA binds to the A site, bringing its amino acid, serine, alongside alanine. Another peptide bond is formed. Then the transfer RNA, which no longer has an amino acid attached, leaves the P site, and the whole ribosome moves along until the transfer RNA with the growing peptide chain occupies the P site, and the A site is adjacent to the next codon on the messenger RNA. We still don't know exactly how all this happens, but we do know that it involves a number of other molecules beside those shown here. These are soluble proteins from the cytoplasm called elongation factors. The process also needs a supply of energy. Watch the sequence again while the next amino acid is added on. The transfer RNA binds to the A site so that its amino acid is lined up with the protein chain. A peptide bond is formed. The previous transfer RNA leaves the ribosome, and the whole ribosome moves along. This sequence of events is repeated over and over again until the protein chain has reached its full length. Each time, one more amino acid is added to the protein chain, and the ribosome moves a bit further along the messenger RNA. In the cell, this process happens very quickly. To make a small protein, one with about 100 amino acids, takes less than 10 seconds. At the end of the information sequence on the messenger RNA, the ribosome meets a termination codon, which may be UAG, UGA, or, like this one, UAA. None of the transfer RNA molecules in the cell has an anticodon to these, so no more amino acids can be added to the chain. Instead, a release factor, another soluble protein from the cytoplasm, binds to the ribosome. This causes the protein chain to be released into the cytoplasm and, of course, ends protein synthesis on that particular ribosome. Usually, though, a number of ribosomes thread their way along a length of messenger RNA at the same time. You can see the ribosomes in this electron micrograph. In fact, there are so many of them clustered on each length of messenger RNA that it's very difficult to see the messenger RNA at all. Complexes like these are known as polysomes, short for polyribosomes, and they enable a number of molecules of the same protein to be produced simultaneously. By making new proteins in this way, the cell is able to grow and divide, keep its structure intact, and carry out all its complex life processes. This is an aerial view of a field of sugar beet. It should be full of healthy green plants, but it's been infected by a virus. Where the viruses have attacked the plants, the leaves have turned yellow, making the crop useless. A different virus has attacked these cattle, producing the terrible symptoms of foot and mouth disease, great suffering for the animals, and possible financial ruin for the farmer. Chickenpox, mumps, measles, even the common cold, are all caused by viruses. Fortunately, their effects are well known, and in most cases not too severe. But the effects of a virus can be devastating, as the concern and publicity about AIDS show only too clearly. Preventing the spread of viruses is not easy, although in some cases it has been done successfully. In this field, for example, the right-hand half of the crop was sprayed to kill the insects which transmit the virus, hence the healthy green plants. The spread of the polio virus is being controlled by the polio vaccine, a safe and effective way of building up resistance to the virus in the population. And vaccination has totally eliminated the smallpox virus, which used to be a major cause of suffering and death. The economic, social and medical consequences of virus infection are obviously far-reaching. So what are viruses? Viruses are very tiny particles, 400 or 500 times smaller than a red blood cell, which can only be seen in an electron microscope. They are made up of nucleic acid and protein molecules. The nucleic acid is either DNA or RNA, depending on the virus, and it's surrounded by a protective coat of protein. The protein coat usually consists of lots of identical subunits, but the way in which these are arranged is characteristic of different viruses. In the tobacco mosaic virus, for example, a virus which infects tobacco plants, the protein subunits are built around a coiled RNA molecule, forming a tube. This is what it looks like when it's magnified several thousand times in the electron microscope. In contrast, another virus, called Adenovirus, which causes respiratory infections in young children, is nearly spherical. This virus contains DNA, and the protein coat packs around it to form a geometrical figure with 20 faces, an icosahedron. The protein subunits of the adenovirus show up well in the electron microscope. It's even possible to see that this virus has some extra spikes of protein radiating out from the coat. These are involved in attaching the virus to cells. Some viruses, like the virus that causes AIDS, have a surrounding membrane or envelope. This is made up of lipids and proteins, with the proteins projecting through the membrane. The electron microscope shows that the human influenza virus also has this type of structure. A virus that looks entirely different from the ones you've seen so far is the T2 bacteriophage. This is one of a family of viruses which attack Escherichia coli, a rod-shaped bacterium that lives in the human gut. The DNA of this virus has its protein coat arranged in the form of a cylinder with flat sides and pointed ends, but it also has a tail section. This is used during virus infection. It helps to anchor the virus to the wall of the bacterium. Viruses flourish by making more viruses, but they can't do this on their own. Making new viruses involves making more nucleic acid, making more protein subunits to coat it, and then assembling the nucleic acid and the proteins correctly. And even the most complex virus doesn't have the biochemical machinery for doing this: enzymes, ribosomes, and so on. A virus can only make more viruses by invading a host cell. For some viruses, the host is a bacterium. For others, it's a plant or animal cell. In all cases, the life cycle of the virus is essentially the same. The nucleic acid from the virus enters the host cell. This nucleic acid is the genetic material of the virus. It contains the code for the proteins of the virus coat and the other proteins necessary for the new viruses to be manufactured and assembled. When it gets into the host cell, the protein synthesizing machinery of the cell is taken over to make these proteins, enough for several hundred new virus particles. More of the virus nucleic acid is made as well, and the new virus particles are then assembled and released from the cell. Viruses which are surrounded by a membrane or envelope, like the AIDS virus, are assembled at the plasma membrane of the host cell and released by budding. So each new virus particle comes away coated with a little piece of the plasma membrane of the host cell. This electron micrograph shows a number of new AIDS viruses budding from a cell. With other host cells, the assembly and release of viruses often cause the cell to burst. A suspension of bacterial cells, for example, has a cloudy appearance, the result of light scattering by the cells. But if a virus is added to the suspension, the cloudiness will clear as the cells are infected and burst open. This agar plate has a uniform growth of bacterial cells on it. If the plate was exposed to a virus, wherever the viral nucleic acid entered a bacterial cell and took over, more viruses would be produced, taking over more cells, until eventually all the cells in that region burst, leaving clear spots. These clear spots are known as plaques, and they provide a very useful way of detecting virus infection in bacteria. Each plaque originally came from one virus particle. Staining the remaining bacteria with a dye makes it easier to count plaques. For a virus to enter a host cell, it must first attach itself to the surface of the cell. It can only do this if the surface has specific receptors for that virus. Consequently, each virus has a very restricted range of host cells. Plant viruses never attack animal cells, for example, and most of the viruses that attack domestic animals can't harm humans. The nucleic acid from viruses gets into cells in different ways. Animal viruses generally enter their host cells intact, and it's only when the virus is inside the cell that its coat is removed and the nucleic acid can start taking over the cell's metabolic machinery. On the other hand, bacterial viruses, like the T2 bacteriophage, usually inject their nucleic acid into the bacterial cell, leaving the protein coat outside. It was the T2 bacteriophage that was used by Hershey and Chase in their experiments to show that DNA, not protein, contains the genetic information. When Alfred Hershey and Martha Chase did their experiments nearly 40 years ago, biologists were not sure how genetic information was carried from one generation to the next. It was known that in eukaryotic cells, the genes are located in the chromosomes in the nucleus. It was also known that chromosomes are made up of DNA and protein, so either could have been responsible for passing on the genetic information. In the case of DNA, its double helix structure hadn't been worked out. It was known that DNA consisted of long chains of nucleotides, but it was thought that the simple repeating structure of sugar, base and phosphate was much too simple to carry the complex genetic message. Proteins, on the other hand, obviously were complex structures, and they occurred in many different shapes and sizes, and with a variety of different properties. So perhaps it was the proteins closely associated with DNA that passed on the genetic message. Hershey and Chase were among the first to answer this question through their experiments with the T2 bacteriophage. They chose this particular virus because they knew that it contained only DNA and protein, and because it readily reproduced itself in a bacterial cell. First of all, they prepared some radioactively labelled T2 bacteriophages. To do this, they grew bacteria in a medium containing radioactive phosphorus, as is happening here. When the bacteria took in nutrients from the medium, they also took in the radioactive phosphorus, which became incorporated into many different molecules in the cells. The suspension of bacteria was then infected with the T2 bacteriophage. To make new virus particles inside the bacterial cells, materials from the cells have to be used. So some of the radioactive phosphorus was passed on to the virus particles. When the virus infection was complete, and the bacterial cells were full of new viruses, they burst, and the cloudy suspension became clear. The very tiny virus particles were separated from the fragments of the bacterial cells by centrifuging. The cell fragments were pulled to the bottom of the tube, leaving the virus particles in the supernatant. This was centrifuged at higher speeds to purify the virus particles. This is the concentrated suspension of purified viruses, and a Geiger counter confirms that the viruses are now labelled with radioactive phosphorus. It's the radiation given off by the radioactive atoms that makes the counter click. Now, a T2 bacteriophage only contains DNA and proteins, and there is no phosphorus in proteins. So the radioactive phosphorus must have been in the DNA part of the virus, in the repeating units of base, sugar, phosphate. Making the DNA of the virus radioactive allowed Hershey and Chase to follow it wherever it went, and they did an experiment to see what happened to it when the virus infected bacteria. They took a suspension of bacteria that had no radioactivity in it, and they added the radioactive virus. They left the mixture for a few minutes to allow the viruses time to attach themselves to the bacterial cells and pass materials into them. If the virus DNA had entered the cells, the radioactivity should now have been inside the cells. To check this, the virus material remaining on the outside of the cells had to be removed. This was done by putting the suspension of cells and viruses in a blender and rotating the blades vigorously for a few minutes. This treatment broke all the remaining virus material off the surface of the cells, and when the mixture was spun in a centrifuge at low speed, the bacterial cells were pulled to the bottom of the tube, leaving in the supernatant the virus material that had not entered the cells. If the DNA from the virus had been passed to the cells during infection, the pellet of cells should have been radioactive, and the Geiger counter showed that it was. To see if there was any radioactivity in the virus material left outside the cells, the supernatant was centrifuged to sediment the material. And this needed to be done at a much higher speed because the virus fragments are very much smaller than the bacterial cells. After centrifuging, the material that had sedimented was tested for radioactivity. It hardly contained any. This experiment showed that when the virus attaches itself to the bacterial cell, its DNA passes into the cell. But it was still possible that some protein passed in too. This wouldn't have been detected because it wasn't carrying a radioactive label. So Hershey and Chase did another experiment to see if any protein from the virus entered the bacterial cell. The experiment was exactly like the one you've just seen, but with one important difference. Instead of labeling the DNA part of the virus with radioactivity, they labeled the proteins, using radioactive sulfur. They could do this because proteins contain sulfur. It's in the amino acid cysteine, for example. DNA doesn't contain sulfur. So, if bacteria were grown in a medium containing radioactive sulfur and were then infected with the T2 bacteriophage, new viruses were formed which had radioactive protein coats. The purified radioactive viruses were then used to infect non-radioactive bacteria. After allowing time for the viruses to pass material to the cells, the protein coats of the viruses were broken off the outsides of the cells in a blender. The cells and protein coats were then collected separately by centrifugation. When the pellets were checked for radioactivity, the Geiger counter showed that the bacterial cells contained very little radioactivity. The protein coats, on the other hand, were highly radioactive. This showed that no virus protein had entered the bacterial cell during infection, and confirmed that the DNA of the T2 bacteriophage must be the carrier of the genetic information. Although Hershey and Chase's experiments only proved this for viruses, in the last 40 years many other experiments have been done which show that in all living things, nucleic acid is the carrier of genetic information.